Shri Gurubhyo Namaha, children. Today we are going on to the last topic of the lesson, optics. Okay, so we are going to learn about microscopes here. Now, you know very well what for we use microscopes. What is the use of a microscope? Any guesses can we make? Microscopes are used to see very tiny objects. They are optical instruments which are used to view very tiny objects. In labs we will go use this microscopes. In biology labs you could have seen teachers having this microscope to view slides. So they are used to view very tiny objects. Okay, so we are going to learn about two microscopes here. One is simple microscope, the other one is compound microscope. Now all these microscopes involves usage of lenses. Since we talked about lenses, so where are these lenses used? The main use of lens is in microscope. Okay, so let us start with a simple microscope. Now here is the diagram, like how the image is formed in a simple microscope. Now this resembles the image formation of convex lens. The last case, in one point alone, in one formation alone, I have told you the convex lens forms virtual and erect image. So image formation in convex lens, last case. So that is the circuit diagram here. Look at this diagram. Actually, this simple microscope, it consists of convex lens. Now look at the convex lens. Where is the convex lens? This is the convex lens whose focal length is very short. It will have a very short focal length. Now this lens is held near the eye so that you get an enlarged image. Now this is the image that is formed due to the convex lens, and this position of convex lens alone will form a virtual and erect image. For all the positions, for all the other positions, convex lens forms a real and inverted image. Only for this position you have a virtual and erect image. So now look at the circuit diagram here. Not circuit diagram, ray diagram, sorry. Now here this is the object, AB is the object. Now this object is placed at a point within the principal focus. The object is kept between O and F. Okay, this is optical center, this is focus. Now you keep the object in between this. Okay, so object is kept here, and you will have your eye, this whoever is observing, it is just placed behind the lens. Now what happens? As per this position of the object, let us take two rays here. One ray is parallel to the principal axis, it passes through the focus, and the other ray passing through the optical center, without deviation it goes. So these two rays, when you happen to see, now these two rays, when you bring it at the back side, it appears to come from this place, which is considered to be the position of the image. Now this forms a virtual and erect image. The image formed will be on the same side of the object, and the distance equal to the least distance of distinction, that is it is less than 25 centimeter. It is equal to 25 centimeter. So this is how the image will be formed. Now the object is small. That is why we go for microscopes. Now look at the height of the object. It is a very small object, right? But look at the image. Now the image formed will be a virtual image. Virtual and erect image, and the size of the image, it is a magnified image. You will be able to see a very enlarged image of the object. That is why we go for this microscopes. As I said you earlier, what for we use microscopes? Microscopes are used to view magnified image of a small object. Okay, now this is the ray diagram for a simple microscope. Okay, and what are its uses? Now uses of simple microscopes are, first uses, they are used in watch repair shops. Okay, watch repairers, They will use this type of lens. Next, microscope. Then to read small letters very clearly, we will go for these types of simple microscopes. Then third one, to observe parts of flower. That's what we use in lab. Parts of flower, insects, etc. can be viewed through this microscope. Okay, and the fourth point is to observe fingerprints. Okay, to observe fingerprints in forensic science. Forensic science department, that's what, suppose if you have to find out any traces of any particular person, you will go for fingerprints which are used which can be seen only through microscope. So these are some of the uses of a simple microscope. Okay, this is a very small topic. Let us go to the next topic, compound microscope. Now, compound microscope, it is also used to view tiny objects. But when compared to simple microscope, this compound microscope has better magnification. You will be able to see the things much more better than a simple microscope. The magnification is, that is the ability to see the images much more better than simple microscope. Okay, now how can you increase the magnification? Means you just change the focal length. Aதாவது, you decrease the focal length of a convex lens which are used, you will be able to have better magnification. Okay, now let us look into the construction of compound microscope. Here you have two lenses, children. Look at the diagram. Two convex lenses are used. Okay, let me explain you the diagram. There are two convex lenses. Now, the lens with the shorter focal length is placed near the object. So, where is the object, children? The object is here, children. AB is the object. Okay. Now, there you will keep a convex lens. Now, this convex lens is supposed to have a shorter focal length. The focal length is shorter, and this lens is called as objective lens. Now, this is kept near the object, right? So, hence this lens is called as an objective lens. Since it is kept near the object and the focal length of this lens is very much shorter, this is called as objective lens, and this is called as an eye lens. Now, this is again another convex lens with focal length having a larger value. Here, the focal length of the convex lens that is fixed here, that is kept here, is larger than this. Okay, and it is kept near the eye of the observer. So, this is also called as eye lens, or otherwise it is called as eye piece. Okay, now let me explain you the first part of the diagram. Listen, this part alone we will take. Now, the object is here. Now, as far as this object is concerned, the object is between C and F. So, where will be the image formed? The image will be formed beyond F. See here, you have to recall the rule that is image formation of convex lens. Which image? Yes, you have to go through the rule, object placed beyond C, sorry, between F and C. When the object is based between F and C, now here the object, this is F, this will be C somewhere. So, the object is placed between F and C. Where will be the image? The image will be formed beyond C. Okay, that is page number 21. The topic is there, when the object is placed between F and C. So, this is how, this is where the image is formed, beyond C. This part alone I am taking first. First part, you keep the object between C and F, the image is formed beyond C. This is the first part of the ray diagram. Now what happens? This will become the object for this lens. Okay, this will become the object for this lens. Where is this position of the object? The object is between O and F. Now, suppose if this is going to be the principal focus, where is the object? The object is between Yeah, O and F, where will be the image formed? The image, this is what the last case we discussed. When the object is within the principal focus, the image formed will be a virtual image, right? So the image is formed here. Okay, so this becomes the object for this lens. Okay, and the image will be formed here, which is a virtual image. The position of the eye lens is adjusted in such a way that the image A dash B dash, where is A dash B dash? Here, A dash B dash will be within the principal focus. Now this eyepiece forms a virtual, enlarged and erect image on the same side of the lens. Now this is the virtual image that is formed. Now look at the object here. The object is very small in size. Look at the image here. Is it clear? So this is the image that is formed, and when compared to simple microscope, the image formed in compound, look at the size of the image here. This is the size of the image in simple microscope. Now compound microscope, you have much more larger image, because as I told you, compound microscope has better magnification. Okay, so it has got 50 to 200 times. See here, 50 to 200 times better magnification than a simple microscope. So to have a very clear image of the tiny objects, we will go for compound microscope. Is it clear? So these are some of the microscopes which are used to view smaller objects. There are some microscope, there is one more microscope called traveling microscope. Actually, this you will be learning in your higher classes. When you come to +1 and +2, we will use this traveling microscope, which works on the principle of 1 year. We will be teaching you that in your higher classes, but you know that also. What is a traveling microscope? It is it is an instrument that is used for measuring very small length. Okay, very small length with minimum accuracy of 0.01 millimeter, where you used to measure the various lengths. We go for traveling microscope. Okay, we'll teach you that when you come to your higher classes. So with this, microscope topic gets over. The next topic is telescope. There is not much more information that is given about telescopes in your textbook, children. Now, anyhow, I will tell you, just listen. What is a telescope? Now, telescope is an optical instrument. Again, telescope, again, it is an optical instrument. Here, we use this optical instrument to view distant objects. Okay. Now, solar eclipse, lunar, last week also we had a solar eclipse. Yes or no? So, those eclipses which are happening in the sky, we cannot see it very clearly through our naked eye. Okay, so we go for these types of instruments. These are nothing but optical instruments again, which are used to view distant objects. So there are so many telescopes, and Galileo Galilei, he was a famous scientist. He was a physicist to make a telescope to view distant objects, distant stars. And there is a physicist named Kepler. He also invented a telescopes to view satellites, rings of Saturn. So many telescopes were discovered. Okay. Now there are two types of telescopes based on the optical property. One is a refracting telescope. The other one is reflecting telescopes. Okay. And astronomical telescope. Astronomy is a study of heavenly bodies. Now, a telescope which is used to view heavenly bodies like stars, planets, galaxies, satellites. In order to view all these things, we go for what is called as an astronomical telescope. Now, terrestrial telescopes are also used to view the image in a astronomical telescope. See, it is not able to view the objects that are on the surface of the earth. They will go for what is called as a terrestrial telescope. Now the terrestrial telescopes, it forms a erect image. Okay. Now these are some of the important telescopes. The topic is discussed in your textbook, children. I want you to go through And the last topic they have discussed about is what are the advantages and disadvantages of telescopes. Advantages of telescopes, you can view elaborately the galaxies, planets, stars and other heavenly bodies through telescopes. And you can also take the photograph by attaching a camera to these telescopes. And they can be viewed even with low intensity of light. Objects, satellite or heavenly bodies can be viewed with low intensity of light using telescopes, right? And the disadvantages are frequent maintenance is needed, and it is not easily portable one. You cannot take it, and it has to be it requires offer maintenance to be given to those telescopes, so that otherwise the instrument may get spoiled. Okay, so these are some of the disadvantages of telescopes. So with this we are completing the lesson optics. Okay, now I am going to discuss some problems which are there in the textbook. Now this problem is a book back problem, solved example. A beam of light passing through a diverging lens of focal length 0.3 meter, 0.3 meter, f is 0.3 meter, appears to be focused at a distance of 0.2 meter behind the lens. At a distance of 0.2 meter behind the lens is it forms an image. Okay, now V is given. So V is given, which is 0.2 meter. Find the position of the object. So what should you find out? Position of the object means, in the sense, you have to find out the value of U. Okay, now diverging lens. What is a diverging lens? Diverging lens means it is concave. Okay, converging lens is convex. So diverging lens means you have to assume that the lens used is concave lens. So as far as concave lens is concerned, the focal length of a concave lens is negative. Okay, and the image formed is also on the same side of the lens. So virtual image will have a negative sign according to the sign convention. Hope you remember this. If you are not thorough with sign convention, you will not be able to understand how this negative and positive signs are coming. Now the values which are measured to the left side of the lens are measured as negative. As far as concave lens is concerned, image is formed on the left side, so you measure the image distance negative, and focal length is also on the left side, so you measure it with a minus sign. Is it clear? Now I have to find out what is the position of the object, which is U. Okay, so what is the formula? The formula is 1 by F is equal to 1 by V minus 1 by U. Okay, I need to find out 1 by U, so that alone bring it to the left side. So 1 by U becomes, rearrange this formula, 1 by V minus 1 by F. Okay, now I have the values for 1 by V. 1 by U will be, what is V? 1 by, now this should have a negative sign. The negative sign is not there. So, so you should have minus 0.2 minus of 1 by minus 0.3. Okay, now when I take the LCM and simplify, 0.2, 0.3 is there, convert it into centimeter, you have 20, 30. When you convert it into 0.2 meter can be converted as 20 centimeter, and 0.3 meter can be converted into centimeter as 30. When you convert and take LCM and simplify, you will have the value of, now I will get the value of 1 by U to be minus 10 by 6. In that case, U will be minus 6 by 10, which is equal to minus 0.6 meter. So U will be minus 0.6 meter. I don't know whether you are whether you have read sign convention. If the sign convention is not clear, the sum will not be clear. Okay, so this is the solved sum. The next one is unsolved. Unsolved page number. Now I will write the page number of this sum. This sum is in page number 30. And the next sum is also in page number 30, numerical problems. Okay. Now I'm going to solve this problem. Check with the Cartesian conventions. An object is placed at a distance of 20 centimeter. 20, u is always negative. So u is minus 20 centimeter from a convex lens of focal length 10 centimeter. So focal length f is equal to plus 10 centimeter. As far as convex lens is concerned, focal length is positive. So it is plus 10 centimeter. Find the image distance. Image distance, you have to find out the value of v and nature of the image. Nature of the image in the sense whether the image formed is real or inverted or virtual or erect. That you have to find out. Okay. So I will write the formula. What is the formula? 1 by f is equal to 1 by v minus 1 by u. Now what is that I should find out? I should find out the value of v. So bring that term to the left side. So I have, when I bring this to the left side, it becomes minus 1 by v is equal to, I will have this 1 by f. When it comes to this side, it comes as minus 1 by f, right? So minus 1 by f minus 1 by u. I take all the negative sign out. So I have the formula to be 1 by v is equal to 1 by f plus 1 by u, removing the negative sign. Okay. Now I substitute the value of f. What is f? F is 10 centimeter plus 1 by, what is u? U is minus 20. Okay. Now take the LCM. When I take the LCM, what will be the LCM? It is 20, right? So here I have 2 plus minus 1 I have here, because minus sign is there down. So it is minus 1. So 2 minus 1 is 1 by 20. Now 1 by v is equal to 1 by 20. So v will be 20 centimeter. So v is 20 centimeter in the sense it is positive or negative? It is positive. So whenever we have the image distance to be positive, in that case, what is the nature of the image formed? The nature of the image formed is real and inverted. Real and inverted image. Are you able to understand? Yes. And we'll find out the magnification also for this. What is magnification formula? Magnification formula is m is equal to v by u. So what is v? V is 20, u is minus 20. So what is magnification? Magnification is minus 1. Is it clear? You understood how the sums goes on? So first I have found out the value of v, then I have found out the nature of the image. Since v is positive, the image is formed is real and inverted image. Clear? Yes? Okay, the next sum. Again, this sum is there in page number 30. Second sum. Okay. An object of height 3 centimeter. So h is given as 3 centimeter, is placed at 10 centimeter from a concave lens. So u is minus 10 centimeter, and they have given the focal length. Focal length of a concave lens is minus, so it is minus 15 centimeter. Find the size of the image. So size of the image in the sense, what is size? Size means you should find out the value of h dash. Okay, right? So first we'll find out v, then we'll find out h dash, right? So what is the formula again? Lens formula, 1 by f is equal to 1 by v minus 1 by u. So I need to find out 1 by v. So 1 by v is equal to, take this to the other side, it becomes minus 1 by f minus 1 by u. Take the minus sign everywhere. So it is 1 by v is equal to 1 by f plus 1 by u. Substitute the values. 1 by f is minus 15. So it is minus 15 plus 1 by u is minus 10. Okay. So this becomes simplified. So you have the LCM to be 150. Minus 15 minus 10, it will be 150. When I take the values up, I will have minus 10 plus minus 15. So this will be minus 10 minus 15, which is minus 25 divided by 150, which is value of 1 by v. 1 by v is minus 25 by 150. What is v now? V will be 150 by minus, let me take it up, by 25. So 150, 6 times it goes. So it is minus 6. So what is the value of v? V will be minus 6 centimeter. So if it is going to be minus 6, then you will have, what is the nature of the image? Virtual and erect. And I will find out the magnification. Magnification will be v by u. Okay, minus 6 divided by minus 10, which is 0.6. So magnification is h dash by h also. So h dash will be m into h. M is 0.6. H is already given. H is 3. So 0.6 into 3 is 1.8 centimeter, which is the value of h dash. Is it clear? So I want you to take down this problem. I'll be sharing the link. Take down these problems. I will try to post some more problems related like this. You have to find out view, V, F, magnification. Only when you practice all these problems, you will be able to attend the sums which is given in the question paper. Okay, the answers for the questions, I will tell you what has to be done. So go through these problems and try to read the entire lesson. Try to mark the answers. I will dictate what has to be done in the next class. Okay, see you children.